Hello everyone, welcome to the channel Microbial World. Today let's learn about transformation in bacteria. Bacterial transformation is a process where bacteria take up foreign DNA from their environment and incorporate it into their own genome. The uptake and incorporation of foreign DNA into bacterial cells leads to genetic alteration and potential expression of new traits such as pathogenicity or antibiotic resistance. The phenomenon of bacterial transformation was discovered by Frederick Griffith in 1928 during his studies on Streptococcus pneumoniae. Griffith observed that non-virulent bacteria could become virulent when exposed to heat-killed virulent bacteria, suggesting the transfer of genetic material. So, Griffith's experiment involved two strains of bacterium Streptococcus pneumoniae. One is virulent smooth strain called S strain and non-virulent rough strain called R strain. He found that when he killed the virulent S strain with heat and mixed it with live R strain, the R strain transformed into virulent S strain. This suggested that some transforming principle from the heat-killed S bacteria was taken up by the R bacteria, making the R bacteria virulent. This was later identified as DNA. Transformation involves the following steps. DNA uptake. Bacterial cells take up extracellular DNA from their environment. DNA integration. Foreign DNA is integrated into the bacterial genome through processes like recombination. Expression. The integrated DNA may be expressed, leading to the production of proteins encoded by the foreign DNA. Bacterial competence refers to the ability of cells to take up exogenous DNA. Competence can be induced artificially through methods like heat shock, electroporation, or chemical treatments. In heat shock method, the bacterial cells are exposed to a sudden increase in temperature, typically around 42 degrees Celsius, followed by rapid cooling on ice. This thermal shock destabilizes the cell membrane, allowing DNA molecules to enter the cell. In electroporation, the bacterial cells are subjected to a brief electric pulse of high voltage. The electric field disrupts the cell membrane, creating temporary pores through which the DNA molecules can enter the cell. Chemical transformation involves treating bacterial cells with chemical agents such as calcium chloride that will disrupt the cell membrane, making it more permeable to DNA uptake. Some bacteria are naturally competent, meaning they possess the machinery for DNA uptake under specific environmental conditions. During transformation, DNA fragments are released from a dead degraded bacterium. These DNA fragments are usually about 10 genes long. DNA fragments released from the dead donor bacterium bind to the DNA binding proteins, that is, transformosomes, on the surface of a competent living recipient bacterium. A nuclease degrades one strand of the donor fragment and the remaining DNA strand enters the recipient bacterium. Competent-specific single-stranded DNA binding proteins bind to the donor DNA strand to prevent it from being degraded in the cytoplasm. Rec-A protein plays a crucial role in the transformation process of bacteria, particularly in facilitating homologous recombination. So, Rec-A protein promotes genetic exchange between a fragment of the donor's DNA and the recipient's DNA. This involves breakage and reunion of paired DNA segments. The donor DNA is integrated into the recipient's DNA, and the transformation process is completed. Thank you for watching.